Hello and welcome. In this video, we will talk about the recent LncRNA profiling technology development and what is the best way to profile the non-coding RNAs. Currently, there are two major technologies for transcriptome-wide profiling, namely microarray for both LncRNAs and mRNA, and RNA sequencing for mRNA. RNA sequencing has many limitations for IncRNA. Other technologies such as qPCR, Northern blot, nanostrands are not transcriptome-wide. Their throughputs are limited to a few hundred genes. The entire IncRNA transcriptome has tens of thousands of transcripts. One of the major characteristics of IncRNA expression is LncRNAs are generally expressed at much lower levels. In the GenCode catalogs of entire transcriptome, the median LncRNA level is about 1/10 of the mRNA. In this quantitative in situ hybridization, FISH, one dot represents one copy of the RNA. There are just a few copies of LncRNA enrolled in the cells, whereas FOXF1 mRNA is present in high copy numbers in the cytoplasm as well as in the nucleus. The other reasons of LncRNA appears to be low in abundance is that LncRNAs can be highly specific and restricted for particular cell types. For example, individual hippocampal LncRNAs are expressed in particular subregions and layers in CA1, CA2, CA3, and dendrite gyres. Even these LncRNAs can be expressed at high levels in the corresponding cell types that can become diluted in the total RNAs extracted from the entire piece of tissue. So what happens if using RNA sequencing for LncRNA profiling? First, a little math about RNA sequencing. RNA sequencing is count-based data. It measures RNA abundance by counting the sequencing read counts. A fundamental numeric properties of integer counts is the intrinsic random Poisson error. This final plot shows the error margins in relation with the read counts by Poisson model. For example, a read count of 5 has an error range between 1.6 and 11.7. The bigger the count, the smaller the error. In real life or RNA sequencing, the actual error is even much worse than the theoretical Poisson model, which is known as overdispersion, as shown in the orange curve. This overdispersion is better fitted by a negative binomial model, which can adjust the Poisson curve, the blue curve, by a shape factor. The point is, with just a few recounts for a gene, even exactly identical RNA sequencing runs can have manifold difference just by random error. You need hundreds of counts for an error less than 20%. This is the principle behind limit of detection (LOD) to detect the presence of a sequence, and limit of quantification (LOQ) to quantify the amount of the sequence. So what is the typical read counts coverage for LncRNA by RNA sequencing? In an RNA sequencing run, the top 1% expressed genes, such as housekeeping genes, occupied about 40% of all sequencing reads. The LncRNAs at the lower expression levels in the gene expression percentile receive very few read counts. Also, RNA sequencing reads rely on being mappable to the reference transcriptome to be counted. For well-established protein coding mRNA, this is not a problem, of a less established LncRNA reference. Many LncRNAs are simply not mapped and thus disregarded. The LncRNA FPKM, or fragments per kilobase per million reads distribution, is shown in the orange curve compared with the mRNA shown in red. LncRNAs are obviously much lower than mRNA in expression levels. Using known amounts of RNA sequence spike in, the minimum counts needed for quantifying the RNA, that is the limit of quantification, LOQ, has been experimentally determined to be 3.13 FPKM. That is, only the RNA expressed higher than LOQ can be quantified. For mRNAs, the majority is above the LOQ, which is okay. For LncRNAs, this yellow shaded area represents less than 5% of the total LncRNA population, which is poor and not okay. A common purpose of gene expression profiling is to compare the differential gene expression of one condition versus another. There is a minimum rate requirement to detect the differential expression in fold change, which is called the limit of detection of ratio, or LODR. As in the spiking experiment published in Nature Methods, the expression levels to detect the fold change and p-values are displayed as limit of detections of ratio, LODR. This gives us an idea that at a p-value cutoff of 0.01, you will need about 1000 counts for 2-fold change. For bigger 16-fold change, next counts at 50 are still needed. In other words, unless for genes with exceptionally large changes, you need large count numbers to detect differential expression. To translate these numbers for LncRNA differential expression analysis, hopelessly tiny fractions of LncRNA, that is the red area under the curve, which is barely visible, have enough counts to see a smaller differential expression at a full 2-fold change. This limit of RNA sequencing for low abundance transcript is well known. In fact, there are commercial kits trying to get around this problem by using so-called targeted RNA sequencing, where enrichments of low abundance RNA by hybridization oligos or PCR amplicon panels are used to get low abundance transcript enriched and sequenced. But by doing enrich then sequencing, you will have to go through the troubles of extra procedures and errors and biases introduced with them. Microarray probes can sensitively detect transcripts as low as 1 transcript per cell. The target sequence hybridization is independent of other unrelated sequences and is perfectly fine even in the presence of other highly abundant RNAs. Physically, the affinity binding enrichment and the intensity measurement steps are simultaneous, which is very simple and straightforward. In this large cohort of clinical study published in PNAS, microarrays, the red curve, show far less error and far better performance than RNA sequencing, the black curve, for transcript at even low levels. For a head-to-head comparison between microarray and RNA sequencing in IncRNA profiling, metastatic colorectal cancer cell line SW620 and non-metastatic primary cancer cell line SW480 were profiled by Arraystar LncRNA Microarrays and RNA sequencing. The RNA sequencing was carried out using Illumina HiSeq 4000 system for paired and 10G coverage, much deeper than regular 6G coverage. The differentially expressed genes was selected using the same stringency cutoffs of FC greater than 3-fold and false discovery rate, or FDR, less than 0.05. In this outcome summary table, close to 2,000 differentially expressed LncRNAs were detected by Arraystar LncRNA Microarray, whereas fewer than 100 differentially expressed LncRNAs are detectable by RNA sequencing. The microarray signals detected the presence of more than 20,000 LncRNAs, whereas RNA sequencing detected only 1,750 LncRNAs. Clearly, microarray is more than a magnitude more sensitive for LncRNAs. The advantage of using microarrays for LncRNA over RNA sequencing is dramatic. For mRNAs, microarrays and RNA sequencing detected about 1,500 differentially expressed mRNAs, so the mRNA performance is similar for microarray and RNA sequencing. Like mRNAs, LncRNAs are alternatively spliced into LncRNA transcript isoforms. These LncRNA transcript isoforms can have distinct biological activities and sometimes even opposite functions. For example, oncogene becomes tumor suppressor. For example, MEG3 LncRNA genes has 10 exons, alternatively spliced into 12 MEG3 LncRNA isoforms. The common exons are colored yellow and alternatively retain exons in other colors. These isoforms have different p53 transactivation activities. Profiling LncRNAs at the transcript-specific level is biologically meaningful. When it comes to transcript-specific profiling, it is very challenging for RNA sequencing, both physically and computationally. The short NGS sequencing rates in the common exonic regions do not provide connecting information for assembling exons. In fact, the top popular transcript mappers, such as HiSat2, MapSplash2, and TopHat2, have sensitivity of less than 10% recall rate. Also, transcript isoforms are not evenly expressed. Major isoforms may receive enough recounts coverage, whereas minor isoforms may not receive enough coverage for quantification. As shown in these diagrams in the middle, LncRNA exon coverage are not sufficient for defining the splice junctions, nor their quantification, whereas mRNAs appears to be okay. The recounts required for transcript isoform assembly is called the limit of assembly, or LOA, which has been experimentally determined to be 23.8 fpkm, using sequencing spiking reference. Very few LncRNA isoforms are only 0.3%. That is, the yellow area under the LncRNA orange curve can be assembled. As observed by Sequencing and Microarray QC Consortium and the work published in Nature Biotechnology, the expression profiling of alternative transcript is particularly difficult for RNA sequencing. And in a large cohort study, 300 million fragments are required for the detections of alternative splicing with 80% of power. 300 million fragment coverage is 10 times steeper than a typical RNA sequencing of 20 to 40 million reads. So to quantitatively profile LncRNA transcript isoforms by RNA sequencing is out of the question. For microarrays, the detections of LncRNA isoform is very easy, unambiguous, precise, and reliable, all the cataloged transcript isoforms can be designed or the probes specific to the splice junction sites or unique exon sequences. The LncRNA array contents are comprehensively collected from all major public genomic, transcriptomic, and specialized LncRNA databases such as GenCo, RNA-seq, LncRNA-DB, FentonCat, Big Transcriptome, NumCo, MyTranscriptome, and IncRNA Wiki. Arraystar's own LncRNA discovery pipeline discovers LncRNA based on all available public sequencing data into Arraystar proprietary transcriptome databases. The LncRNAs are quantified by their full-length status, completeness in 5' and 3' ends, and the protein non-coding potentials by multiple algorithms and experimental evidential supports. The LncRNA collection on the microarray is by far the most comprehensive and the best vetted LncRNA collection than any publicly available LncRNA repositories. The specifications of LncRNA microarrays are copied from our website. For human, there are about 40,000 LncRNAs, among which more than 8,000 are gold LncRNAs, which will be explained in the next slide. On the same chip, the entire protein coding mRNAs are included, which provides simultaneous profiling of coding and non-coding gene expression. As a whole, LncRNAs are less known than proteins. Nonetheless, growing numbers of LncRNAs have been studied in more detail. These functionally studied, experimentally supported, well-annotated LncRNAs have at least one PubMed paper describing them. That thus the gold LncRNA collection tier as low-hanging fruits, having more information ready for follow-up studies. All other LncRNAs are in the comprehensive, high-confidence, reliable LncRNA collection tier. The reliable LncRNA collection tier has been qualified with the transcription unit models to ensure they are in fact transcribed from gene structures, not just random degradation products in data dumps. In addition to the gene expression values, the annotation and data analysis are essential, integral parts of the LncRNA expression profiling to get important insight into the profiling data. Based on 2017 Nature paper, at the topmost level, LncRNAs are classified into LncRNA made from promoterless enhancer regions, that is, E-LncRNAs, and LncRNAs transcribed from promoter regions as P-LncRNAs in the epigenetic context. The LncRNAs are further classified by their genomic context with the nearby protein coding genes as intergenic, divergent, antisense, sense-intronic, for implications in cis-regulating their target genes. The full-length status of a LncRNA is indicated whether it has a complete 5' end as supported by cage sequencing or cap analysis, gene expression sequencing, complete 3' end, or complete full-length at both ends. Some LncRNAs have small open reading frames with small peptide coding potentials are scored. These SM LncRNAs may produce bioactive small peptides. The subcellular localizations in the nucleus or in the cytosol can help to predict LncRNA functions. A nuclear LncRNA is more likely to be involved in the nuclear functions such as transcription or RNA processing, whereas cytosolic LncRNAs is more likely to play cytoplasmic functions such as regulating mRNA translation or competing endogenous RNAs for microarrays. Certain important subgroups of LncRNAs, such as competing endogenous RNAs or cRNAs and antisense RNAs, are especially grouped for their predictable target genes by base pairing with the target sites. cRNAs regulate microRNA-mediated gene silencing by competitive bonding to microRNAs. Natural antisense RNA regulate gene expression by mRNA masking, interference, genomic imprinting, and guided methylation. Highly conserved or ultra-conserved TUCRs across species may underlie vital functions indispensable for the organisms. Non-intergenic RNC LncRNA can act as enhancer RNAs that can impact the transcription at a long distance in cells. LncRNAs that are known to be associated with disease, biological pathways and processes, or highly specific expression in particular tissues are compiled and annotated. The schema of annotation tables provides a wealth of information which will greatly facilitate your LncRNA studies. The new annotations in newly released LncRNA microarrays are indicated as new. Arraystar LncRNA microarrays have been a driving force that have helped research laboratories in the exciting studies of LncRNAs in biology and disease. Several hundred high-impact papers using Arraystar LncRNA microarrays have been published in the past. In summary, to profile LncRNA expression, the LncRNA microarrays are the best choice, much superior than RNA sequencing in terms of detection sensitivity, quantification accuracy, transcript-specific reliability, and a wealth of annotation and analyses. RNA sequencing is inadequate for LncRNA profiling. The word next-gen was more than a decade ago. Nowadays, RNA sequencing is neither novel or next-gen. So do use the best tool for LncRNA profiling job, which is LncRNA microarray. I think I will stop here. Thank you for your interest and enjoy visiting our website.